This is a continuation of the metabolism of glucoses. Pyruvate, the end product of glycolysis, can be further oxidized to form carbon dioxide and water under aerobic conditions. It can be reduced to form lactate, and it can also form the molecule of ethanol, these two under anaerobic conditions. In this reaction, when glucose is in anaerobic conditions, in the presence of some yeast and some microorganism, glucose will undergo glycolysis in the absence of molecular oxygen to produce the molecule of pyruvate, which will undergo an oxidative decarboxylation to produce acetaldehyde. This reduced coenzyme NADH is one of the products of glycolysis that is reinvested to reduce acetaldehyde to a primary alcohol. That's the reason why NADH, neither pyruvate, are part of the overall reaction. This is a method to recycle the reduced coenzyme, which is being transformed into the oxidized coenzyme NAD+. When the rate of reduction of molecular oxygen to water and the rate of the citric acid cycle is slower than the production of pyruvate from glucoses, pyruvate can undergo reduction of the ketone group to form an alcohol group in the molecule of lactate. This is using the reduced coenzyme that was produced during glycolysis, therefore is not part of the chemical reaction. This lactic acid formed in the muscle can be recycled and can be reused by the liver. This lactic acid is going to be part of the Codec cycle, being sent to the liver where it can be metabolized when the amount of oxygen is abundant. Glucose is the main source of energy for the brain. When there are no glucoses in the diet, glucose has to be made from other sources. This process is called gluconeogenesis. In glycolysis, 3 steps of reactions were catalyzed by kinases. These kinases were in charge of phosphorylating carbohydrates. These steps will be now overcome by the use of phosphatases. There is not a production of energy during the process of gluconeogenesis. In this process, the main goal is to produce glucose units from no glucose sources. Some steps that were using kinases during glycolysis now will need to use phosphatases. Glycerol, product of the hydrolysis of triglycerides or fats, can enter this process as dihydroxyacetone phosphate. Proteins can enter as some amino acids as pyruvate or oxaloacetate, and they will go through the same reactions of glycolysis that are reversibles. All others that are not reversible will need to use a different kind of enzyme. Gluconeogenesis will be highly active when the person is under starvation, in a famine, or when fasting. Glycolysis will be highly active upon ingestion of the foodstuff. The process of glycogenesis is the synthesis of the polysaccharide glycogen. Glycogen is a branched polysaccharide synthesized in the liver using units of glucoses. However, these glucose units must be highly activated with phosphoryl group in order to be used to make glycosidic bonds alpha-1,4 and alpha-1,6. During the first step of glycolysis, glucose gets a phosphoryl group on carbon number 6. It will isomerize to fructose 6-phosphate to proceed with glycolysis. When the cell is charged with ATP molecules, the isomerization will take a different route by changing the phosphoryl group from carbon number 6 to carbon number 1. Once it's phosphorylated in carbon number 1, an activated nucleotide, uridine triphosphate, will assist the reaction endergonic to become exergonic to produce the molecule of glycogen. Important to understand also that excess of glucose can be transformed into the formation of body fat. The process of glycogenolysis is the opposite, in which a polymer of glucose, glycogen, gets hydrolyzed to make free units of glucose 1-phosphate that can further produce molecules of glucose. This process is important to keep a constant level of sugar in the blood, which is assisted by the hormone glucagon.